Quantitative real-time PCR, or qPCR, is a technique used to detect and measure a specific DNA sequence in a sample. Unlike conventional PCR, where the amplified DNA is usually analyzed after the reaction is complete, qPCR monitors DNA amplification after every cycle using fluorescence. The reaction still follows the usual PCR steps. Denaturation separates the DNA strands, primers bind during annealing, and DNA polymerase extends them to make new DNA. Ideally, the amount of target DNA roughly doubles with each cycle. As more DNA accumulates, the fluorescent signal increases, producing a characteristic amplification curve. A threshold is set above the background fluorescence. The cycle at which the signal crosses this threshold is called the CT, or cycle threshold, value. A low CT means the sample started with more target DNA, while a high CT means it started with less. There are two common ways to generate the fluorescence. SYBR Green is a dye that fluoresces when bound to double-stranded DNA. As more PCR product is formed, more dye binds and the signal increases. TaqMan uses a sequence-specific probe carrying a fluorescent reporter and a quencher. While they are close together, the quencher suppresses the signal. The probe binds between the primers, and during extension, the 5' nuclease activity of Taq polymerase cleaves it. This separates the reporter from the quencher and produces fluorescence. If the starting material is RNA, such as viral RNA or mRNA used for gene expression studies, it is first reverse transcribed into cDNA. This is commonly called RT-qPCR. qPCR is widely used for pathogen detection, measuring gene expression, and determining the amount of specific DNA or RNA targets in a sample.